We will wait 5 more minutes for people to join.
Radhika, are you on campus?
Yes.
I haven't been on campus in so long. How is it, like a ghost town?
You do see people walking around now. Depending on the time, but it's definitely more like a summer feel. If that
Makes
helps.
sense. Yeah, I know undergraduate researchers just started coming back to campus. I've only been going to campus to get my proactive community testing, the saliva, COVID testing. And I noticed that line is starting to get long. So that was good to see people coming out and doing their testing, and yeah.
Yeah, it's definitely fewer people around, but you do see people. So.
Yeah. I feel bad. I used to, like, in the fall and spring when all the students came back, I was like, oh no, it's so crowded now. And I'm like, I want to be in the crowd. I want to see all the students. I miss the activity going on.
Yeah, I totally get that. Well, trying to make do with whatever we have.
Yeah, absolutely. What building are you sitting outside near?
So I'm in Welch, the chemistry one. So we have a patio. Thankfully, there aren't people around, so I can take off my mask in the outside and stuff.
It's actually really nice to see, like, a real outside background and not a fake Zoom background.
Emily's here too. Nicer.
All right, let's get started. Hello, everyone. Welcome to the third Student Sensory Seminar Series. My name is Zeong Lee. I'll be the chair of today's talk, and I'm also one of the members in the student-led research interest group. The primary aim of our sensory group is to share research in the area of sensor and sensing technologies. We hope the student seminar series will enable knowledge share and foster collaborations. Please visit our website to explore more about our organization. If you would like to present your research or interested in joining our group, please do not hesitate to email us. The title of today's talk is Fluorescent Probe for Imaging Zinc Metalloproteins in Cells. We're very excited to have Dr. Radhika Mehta here. Dr. Mehta is currently a postdoc in Professor Emily Q's lab in the Department of Chemistry at UT Austin. She also completed her PhD degree in the Q lab in 2020. Dr. Mehta obtained her bachelor's from St. Stephen's College in India and master's from Oxford University in England. During her time at UT, Dr. Mehta served as a safety officer for QLab. In addition, she also associated for the Texas Venture Lab Practicum and Texas Health Catalyst, and outreach chair for Association for Women in Science at UT Austin. She is particularly interested in studying metals in biology through chemical and biochemical approaches. She'll be transitioning to a second postdoc in the Merchant Lab at UC Berkeley in 2021. Before the talk begins, I'd like to remind you that you can ask questions at any time by using the chat box. We'll collect the questions and ask the presenter at the end of the talk. Now, I would like to hand over to Dr. Mehta, and the time is yours.
Hi, everyone. Thank you so much for the invite and for the introduction. Great. So, yeah, first of all, happy Mole Day. It's October 23rd. So we chemists like to call it Mole Day. And I'm going to tell you a few stories and just whatever we've gathered so far but in the field of metals in biology, specifically looking at zinc metalloproteins, with the overall goal of exploring and tracking metallation status. And for this purpose, we've been making fluorescent probes, which basically act as our sensors. And so in the K-Lab, we like to think about metals existing in biology in sort of two main forms. First is this labile metal ion pool where the metal is freely floating around in the cell. It's bound to small groups, chelators, and loosely bound and can be stored with storage proteins and with chaperones. And the other pool that we like to think about is this metalloprotein pool, wherein the metal is tightly bound to the protein in question and responsible for the protein's function. Now, one of the things is why should we even care about these metals in biology, right? Why do we want to look at them? And some of the main reasons is an imbalance in this metal ion pool as well as the metalloprotein pool has been indicated in various diseases. Even diseases like Alzheimer's is associated with an increase of zinc and iron and copper in the brain. Diabetes is associated with a zinc imbalance as well. And so to look at both of these labile as well as the tightly bound pool will allow us eventually to think about metal homeostasis in the context of diseases. But in what our approach towards this has been to look specifically and more rigidly at metalloproteins, for the sole purpose that there have been multiple fluorescent probes and sensors developed to track free metal, but there are very limited ones developed for metalloproteins. One of the main reasons being that each metalloprotein is different. And that's why one needs to develop sensors specifically for each of these different metalloproteins. And so our overall goal has been to study in zinc fluxes, so the effects of increasing cellular zinc, decreasing cellular zinc on all these metalloproteins. Where are they getting these metal from? And kind of tightly controlling them is basically been the main purpose of a lot of our work. And the way we're testing this is by developing fluorescent probes, which we then look and use for live cell imaging of these metalloproteins. Our two main criteria for our probes has been A, we want them to interact reversibly, and reversibly only with the metal form of these proteins. So we want to track only the metallated forms of these proteins. And the second form of criteria is, of course, that we want it to be selective so that we're tracking the metalloprotein in question. In my work in the K lab, we've looked at two main metalloproteins, zinc metalloproteins. The first one is the mammalian carbonic anhydrase, and the second one is a bacterial one, Nadelli metallobeta-lactamase, that is actually a collaboration. And I'm going to very briefly talk about what we've seen with carbonic anhydrase, but most of my talk is going to focus on the NDM. And just to give you a brief on the scale of things that we work on, we think about the metals existing as well as the metalloproteins, but also look at the scale and interactions at the cellular level. So much of our time is spent on this fluorescent probe design, which I'm not going to go into much detail today. But we also think about and look at parameters of where these metalloproteins are, how much are they expressed, and those are also criteria that come into the overall project design. So to kind of go delve further into the fluorescent probes that we've made, we have had, as I said, we have had two main things that we want these probes to do. First is we want the fluorescence turn on only when it binds to the active site in the protein in question. So this is sort of the off state and this is the on state. And the second thing is we want to have a reversible selective binding to the holoenzyme. By holoenzyme, I mean the enzymes that contain the metal in them. So we want this fluorescence turn on to only happen when the protein has the metal in there. And so for a fluorophore end, we have two ends. The fluorophore end, we use this class of environment sensitive fluorophores that have this important property that we're harnessing, that is in polar environments like the aqueous media or a buffer, these are basically non-fluorescent. But when we go to a non-polar environment, we see a fluorescence increase. And this non-polar environment is quite similar to a protein pocket. A protein pocket is definitely more hydrophobic, more non-polar than the water surrounding it. And that's what we're harnessing in our design. We expect that once the probe goes and binds and the fluorophore is in the active site pocket, we should see an increase. And the way we are making these probes bind is by modulating the metal binding group, which is denoted here. For carbonic anhydrase, we've used these well-studied sulfonamide groups, and I'm not going to go into too much detail here, but for NDM, we have taken inspiration from these captopril and other inhibitors that are more thiol-based. And so just to kind of go briefly into what we've seen with carbonic anhydrase. So the reason we used this as a first was it's a model system. It's the first zinc metalloprotein that was discovered and studied in the 1940s. And why should any of us care about it? Well, it catalyzes this reversible conversion of carbon dioxide to bicarbonate, and thus it's essential for respiration. It's essential for pH balance in our body and for transport of ions. And without it, most of our cells are not going to work. Our survival and is based also on carbonic anhydrase. But one of the other things is it's also overexpressed in cancer cells. And that's why it's an interesting target to study. Now, what we've seen is we developed this fluorophore, and if you want to get more detail, you can go to see this paper, but we developed this fluorophore that gave us a 12-fold fluorescence response with carbonic anhydrase. Additionally, it bound at low micromolar affinity, which is very similar to how a lot of these inhibitors and probes for carbonic anhydrase bind. And we also tested its selectivity by comparing it with a range of metalloproteins, as well as we did some in-gel studies with it and looked at reversibility, which I'm going to go into in the next slide, and additionally the cell permeability. So when we actually use these probes, especially probe 1A in cells, and this is imaging in HeLa cells, so HeLa is cervical cancer, what we see is when we added probe 1A, we see this fluorescence increase. And that actually decreased when we added a known carbonic anhydrase inhibitor, which would ideally replace out our probe. And this was one of the ways we were able to determine that we are selectively and reversibly using our probe to bind to carbonic anhydrase. This here panel just shows a control where we saw that the free zinc, which was stained using an FC3, a commercially available zinc fluorophore, is not really affected by acetazolamide or AZA treatment. And so having done these, we went on and looked at zinc perturbations with these. And what we saw is when we decreased the free zinc in these cells, 1A or CA fluorescence was not affected. When we increased the free zinc in these cells, again, the CA fluorescence was not affected. And so what I want you to I want you to think about is, and I'm going to tell you why towards the end, is TPEN and zinc parathion, which basically decrease and increase the cellular zinc, had no effect on carbonic anhydrase. So with that, I'm going to quickly tell you about what we've seen with NDM instead. So NDM, why did we want to study it? It was one of the proteins that is studied and identified only in the last decade, and it's been implicated in growing rise of antibiotic resistance all over the world. And so what we've seen with why it's important is NDM is also a zinc inhibitor, a zinc enzyme, and it catalyzes this conversion of beta lactams in last resort antibiotics like ampicillin, like penicillin, and thus makes them ineffective, which gives rise to the growth of bacteria and thus antibiotic resistance all around the world. In the context of what we study, it was an interesting one to study because the NDM methylation status is slightly different from carbonic anhydrase. It has two zincs, as well as its localization is also slightly different. In a bacterial cell, it's known to be expressed in sort of between the outer and the inner membrane. And one of the reasons why this is an interesting target to study for zinc homeostasis is because a lot of the forms of NDM are evolving to becoming active in low zinc environments, which is basically why I'm interested came in here. And so we want to develop these fluorophores that can now target this active form of this protein, because actually there's not been any fluorophore developed so far that can actually track the reversible binding to the active form, the di-zinc form. And so quite similar to what we've done with carbonic anhydrase, we used the same fluorophore scaffold, but our metal binding group here was inspired by thiol derivatives like the captopril here, which are known inhibitors. I won't be showing the structure just because we're preparing this project, this paper right now. But quite similar, we looked at the fluorescence turn on, wherein we saw 12-fold to 17-fold. And the reason I say it is because with NDM1, we saw 12-fold, but with another stronger NDM, stronger zinc binding and NDM form, we saw up to 17-fold. And this could interact and inhibit these proteins at about similar range as these decaptopril ones, as well as selectivity panel that we've shown here, and reversibility and cell permeability. And so I'm going to briefly show what we've seen in cells is, so when we express these bacterial cells with that, so without NDM1, we did not see any fluorescence, which was one of the best ways for us to determine selectivity. Secondly, once we started expressing, we start seeing a lot of fluorescence here, and just a zoom in view shows where these are sort of localized. And so we went on and did sort of similar treatments. What would happen when we treat it with an inhibitor of NDM? What would happen if we treat it with zinc chelators? And so with the inhibitor DL-captopril, we start seeing that decrease, and we were able to actually quantify it here pretty well. So you can see these dots actually start disappearing here. With zinc chelators, both TEEP and here, now at the same concentration as 10 micromolar, we're seeing a pretty significant decrease, which again is much more pronounced at higher concentrations. And here, what we treated it with another zinc chelator, but this depletes the zinc outside the cell. And even that is now showing effects here. So basically, with NDM, now we're able to see that zinc chelators can significantly reduce the fluorescence. And so in the last minute or so, I just want to tell you why we think this is it, and what we've able to gather from these fluorescent probes and sensors. So we saw that with carbonic anhydrase, the effects were basically minimal on because of these zinc perturbations. But in NDM, these were way more pronounced. And one of the main reasons being that the zinc is actually bound very, very strongly in carbonic anhydrase, whereas in NDM, these are much weaker at nanomolar or a micromolar for the second one. Secondly, the active site is much more deep in carbonic anhydrase, whereas in NDM, it's more surface exposed and flexible, which allows a lot of these small molecules, chelators, to come and take it out. And so what we've seen is zinc perturbations practically have no effect in these carbonic anhydrase, but they do in NDM. And this is also one of the interesting ones that I'm going to allude to, because carbonic anhydrase is an essential protein. It's present in all of us. But the NDM one is actually an evolutionary one. It's how proteins in bacteria are evolving to overcome antibiotics. And this sort of ties into to sort of the evolution of metalloproteins as well. But what the future holds for a lot of this work is thinking about what will be the trends at when if we have zinc affinities that are between the micromolar and the picomolar level. What would happen when we have, say, deeper active sites, but a much weaker binding affinity, unlike carbonic anhydrase? And what these would eventually do is allow us to study the interplay of a lot of these metalloproteins with with the labile metal pool, which will eventually help in developing metalloprotein targeted therapies. A few other things would probably be using these probes in diagnostics, in developing fluorescence assays, and eventually also in inhibitor design and testing. With that, I would like to acknowledge my supervisor, Emily, who's been a great support, and it's been a great learning experience. What I actually feel very lucky to have done my PhD here, because I've learned a lot from her, from the other members, through our collaborators, as well as all my doctoral committee members. And so with that, I'd be happy to answer any questions that y'all have. Thank you.
Thank you very much for your amazing presentation. Okay, let's wait a couple of seconds for the questions to come in.
Radhika, do you have ideas for metalloproteins that lay sort of between carbonic anhydrase and NDM in terms of their active site depth or their zinc affinity?
That's a good question, and I think they will vary. There can be metalloproteins that say have in between affinity, but say a deeper active site like carbonic anhydrase or a weaker active site like NDM. And that's actually dependent on the substrate that they work on, or how essential are these proteins. So like carbonic anhydrase is such an essential protein that, in biology, or thinking about it, nature would probably want to conserve it. It would not want small perturbations to affect it, because then in that case it starts affecting our evolution, our entire being. And so I think one could actually probably start off with making different mutants of carbonic anhydrase, which bind zinc weakly, and then see what are the effects there. And that would probably be one of the first steps, I think.
Radhika, you mentioned diseases such as Alzheimer's, diabetes, and cancer. And so I'm assuming your work is kind of to help therapeutics with those diseases. Have you, because of the world that we're in right now, have you thought about the coronavirus in any way?
So that's a great one. So the ACE2, one of the proteins that actually binds to the spike protein of coronavirus, is actually also a zinc protein. So I think one of the thoughts was, okay, can we develop probes for that and track it? We haven't yet. So actually, the L-captopril, one of the molecules that I showed, is an ACE inhibitor. It's a different form of the same protein, but there are very few that have been developed for ACE2, and so I think one of the things which Emily could probably do and think about is developing fluorescent probes for ACE2 as well, and that would allow us to sort of track that in terms of the coronavirus and other proteins that interact with ACE2 as well. So this is probably going to be more towards a diagnostic purpose, but then again, you can eventually use it for multiple different things.
Okay, I have a question. What are the challenges you faced when designing these fluorescent probes?
Okay, those are quite a few. So actually, I think I might have an extra slide on it. Just a moment. Okay, so there are a few different things. One needs to think about the protein in question and how this probe is actually interacting. And so these are some of the factors that we consider and we're going towards is, so we want an ideal fluorescence turn on, which is dependent on the hydrophobic environment of the protein. And each protein will have a different hydrophobic environment. It's also dependent on the electrostatics of the protein, and as well as the sterics that are present there. And so, and the second thing is, of course, the binding affinity, wherein the metal binding group is an important factor. So what we're trying to do now is actually combine both of them and look at it by computational methods like probe docking, which will allow us to see where these probes are actually sitting, where the fluorophore end is, and how is that environment there. And so the challenges are quite a few. but we try and take it step by step. We try and modify multiple binding groups. So I just show you one of them, but we actually have made like a suite of 20 to 30 molecules for this purpose. We also do modifications to the fluorophore and then use it. So I would say it's sort of a few different factors which we try and play with and use computational methods to get a handle on it.
Okay, I see. Do anyone have any other questions? Okay, so if your question is not answered or you have any further questions, please email us. We'll deliver your question to Dr. Mehta and get back to you soon. So I'd like to thank you again for your great presentation, and I wish all the best in your future position in Berkeley. And I'd like to thank our audience as well for joining us today. Give me a minute to share my screen.
Thank you.
Okay, our next talk is molecular sensors and molecular logic gates given by Dr. Adam Sedgwick. We're very much looking forward to seeing you in 2 weeks. Thank you. Bye.
Thank you for the talk.
Thank you for having me. This was